Hello, everyone. Sorry for keeping you all waiting. Welcome to the first student seminar series. My name is Zhang Li. I will be the chair of today's talk, and I'm also one of the members in the student-led research interest group. The primary aim of our sensory group is to share research in the area of sensor and sensing technologies. We hope the student seminar series will enable knowledge share and foster collaborations. Please visit our website to explore more about our organization. If you'd like to present your research or are interested in joining our group, please do not hesitate to email us. Today, we are very excited to have Emma Palmer here. Emma is a MS PhD student in the Environmental and Water Resources Engineering program in the group of Professor Mary Jo Kirisits. She completed her bachelor's degree in environmental engineering at Duke University in 2019. Additionally, Emma holds a chemistry minor and a global development engineering certificate. In 2020, she was selected to be a fellowship recipient through the National Science Foundation Graduate Research Fellowship Program. Her current research efforts are focused on optimizing methods to detect community-scale prevalence of COVID-19 through wastewater epidemiology. Before the talk begins, I would like to remind you that you can ask questions at any time by using the chat box. We'll collect the questions and ask the presenter at the end of the talk. Also, the talk is being recorded and it will be available on our YouTube channel. Now, I would like to hand over to Emma, and the time is yours.
Thank you. So give me just a second. So I'm going to share my screen. Can you all see that?
Yes.
Yeah. Okay.
Yes.
Perfect. Well, thank you so much for the introduction, and thanks for letting me share my research with you. So like was mentioned, I am going to be sharing about how we're using wastewater to detect community scale COVID-19 prevalence. And I'm sharing on behalf of the Canary team, and I'll explain a little bit more about that as we move forward. So first, I just kind of want to get us all on the same page. I'm sure that some of you have heard a lot of details about SARS coronavirus 2 on the news and in popular media, but I just want to review some of that information. So what is SARS-CoV-2? It stands for Severe Acute Respiratory Syndrome Coronavirus 2, which causes coronavirus disease 2019 or COVID-19. So it was a novel human coronavirus that was first reported in China in late 2019. And we have seen previous coronaviruses, including SARS-CoV-1 and MERS-CoV, which were a concern in 2003 and early 2010s, but these were a little bit more isolated. They didn't become a pandemic like we are now experiencing with COVID-19. So COVID-19 was classified by the World Health Organization as a pandemic on March 11th, which it's hard to believe, but that was 6 months ago, and it doesn't seem like it's going away. And so this map on the right here, some of you may have previously seen it, but this is a map tracking the cumulative COVID-19 cases by Johns Hopkins University. And as you can see, it's very widespread, and the US still has a crazy number of cases. And I pulled this map on Monday, so it may be a little bit dated, but I think the big takeaway here is that this disease isn't going away, and we're going to have to learn how to live with it and how to monitor it and how to respond when we see a spike in cases, especially as in the last couple of weeks, even places in Europe that previously thought that they were kind of over that hill, if you will, are seeing a second spike in cases. So I'm just going to go over some basic biology. So think back to your high school biology classes. So pictured here is the SARS coronavirus 2. And so a virus is not like a bacteria. It's just a package of genetic information with some proteins, and in this case, some lipids that package this genetic material. And so to replicate, viruses have to use a host cell machinery to be able to create more virions. And to do this, all viruses go through a very common set of replication mechanisms, and SARS-CoV-2 is no different. So the first thing it does is it attaches to receptors in the human cell. And it is important to note that these receptors exist throughout our body. We originally thought that SARS-CoV-2 and COVID-19 were just a respiratory disease, but these receptors actually are present throughout our body. And that's why we've kind of evolved to thinking of COVID-19 as a vascular disease. And something of interest to us as people who study wastewater is that these receptors exist in our GI tract, which really enables us to track this virus and the shedding of it through our waste. And so, as I mentioned, this virus has to use the host cell machinery to be able to copy itself. And I won't go through all the mechanisms of this. It can get pretty complicated. But I guess the two things I wanted to highlight here is that once it enters the host cell, our cells will replicate the RNA, and then it helps the virus produce the proteins that are needed to package this RNA up and release virions. So these proteins in the RNA itself can be used for detection of SARS-CoV-2. So here is just an overview of the SARS-CoV-2 complete genome. So SARS-CoV-2 is made up of 30,000 nucleotides. So that's our A's, C's, T's, and U's that make up the genetic code. And if you compare this to the human genome, we are made up of a little over 3 billion nucleotides. And so this virus is 100,000 times smaller genetic code than us, but it's wreaking havoc on our world. And I think what I wanted to highlight here is that this genome encodes proteins, 4 major proteins, S, E, M, and N gene, and then some other accessory proteins in these open reading frames that indirectly impact the infectivity of the virus. But what our research is focused on is this N gene, which includes the nucleocapsid proteins, which help to package that RNA into the virions, which in the previous slide were shown as these blue dots. So when you think you're sick or when you go to the doctor to get a SARS coronavirus 2 test, what does that look like? Well, first of all, I just want to highlight that testing is voluntary. It's on an individual basis, and we typically think of it as being reactive as opposed to proactive. So even though UT and other organizations are doing proactive testing to limit the spread of the virus, typically most of the testing is done because you feel sick, you go to a doctor, and then you wait for your results. So can be several weeks of lag time between when you're actually infected with the virus and when you get positive results back. And these tests come in 2 major classes. The first is an antibody or serological test. And we won't talk too much about this because this just tells you, have you recently had the infection? It doesn't tell you if you have an active case of the virus. And this test works by detecting antibodies that indicate that your body has had an immune response against the virus. The other type of test that we'll talk about more extensively is called a diagnostic or viral test. And this basically aims to determine if you currently have signal of an infection from the virus. And the target, as I mentioned in the previous slide, can be antigens, which are those proteins, or it can be the RNA of the virus. And the way that we detect RNA in a sample is first we have to isolate that RNA. So cells are made up of so many different things, including those proteins and lipids that I mentioned earlier, and we have to isolate just the RNA molecules that we want. And then using targeted primers, which in our case are targeting that N gene, we amplify our RNA, and then that amplification allows us to determine how many copies of that and if we see signal of the virus. So because these are reactive as opposed to proactive, something that we were really looking to determine is there any way that we can determine if there's a spike in cases before people go and get tested because of that lag time between when you feel sick and when you go to the doctor and when you get tested and when you get your test results back. And so we call ourselves the canary team. And this is an analogy to when in coal mines, people used to take canaries down to the mines and they would know there's a problem with the air quality when the canaries stopped singing. And it was just kind of a heads up of, hey, you should probably get out of there because it seems like there might be a problem. And so in looking at this, what we've discovered and what scientists around the world have been discovering is that actually wastewater is a perfect tool to use to give us that early warning system. So this was a study done in Connecticut, and as you can see with this red curve on the left, this is copies of the viral RNA per milliliter of wastewater. And as you can see, it has a 1 to 2 week leading indication of the COVID-19 new cases. And so because we know that the virus is shed through urine and stool in pre-symptomatic and asymptomatic and symptomatic COVID-19 positive patients alike, it's a very useful indication as to if there's COVID-19 prevalence within a community. So that comes to our wastewater-based epidemiology work. So wastewater-based epidemiology basically works as a pooled sample, and it's not as voluntary as going in and giving an individual test, because we all go to the bathroom, we all flush the toilet, and every time you flush, you contribute to our sample. And this approach, as weird as it may seem, has been used for a very long time, previously, especially to detect viruses like polio. And so the way that our system works, we have 3 main sample types that we collect. The first is within the conveyance system. So this is the pipes that when you flush your toilet or use the sink, that's where your water goes to be transported to the wastewater treatment system. And so far, these samples have just been grab samples. So that just means we're just taking a cup and basically taking a sample of what's coming out at that time. But we are hoping to transition to what we call composite samples, which represent the input into the conveyance system over a longer period of time. So the other 2 samples that we're getting are within the wastewater treatment plant. And the first is an influent sample, which is just preliminary treatment, which just means that we're removing some of that large debris that would get in the way of our sample collection and processing downstream. And then the second type is primary clarifier effluent. So within a wastewater treatment plant, which I have the schematic on the right, we have several different layers of treatment, and primary clarification basically just settles out some of those larger solids. And both of these sample types within our wastewater treatment plant are what we call 24-hour flow-weighted composite samples. And this is a crucial step in our process because, as I mentioned before, if you just take a grab sample, you just happen to get those people that are contributing to that sample in that one instant in time. Whereas doing this, we can average out and see basically the whole population over 24 hours that have contributed to the sample. However, this is kind of an unwieldy process because we have to measure our flow and ensure that when the sample is taken, that the sample that we get is proportional to the amount of wastewater that's coming into the plant at different times. So this is an overview of our process once we get our samples back to the lab. It's very complicated, and I don't want to get into the weeds too much about this, but I think the basic takeaway that I want people to take away from this is that it's a very time-consuming process. Once we have our sample, then we have to aliquot it, we pasteurize it so that there's not as much risk of infection to the researchers, and we're concentrating on this pellet, so the solids that we obtain after centrifugation of our sample. And then from here, we have to do an extraction process to isolate that RNA. And then we're using polymerase chain reaction, as I mentioned before, to understand and quantify how much SARS-CoV-2 RNA is in our sample. So I'm just going to show some preliminary results. We've been doing a very deep dive into the methods to ensure that we're getting a representative signal for what's being passed into our wastewater. So it's not necessarily, we don't have as much data as we will very soon. And we're very excited to start going through all of the samples that we have. So just here, we have plotted in blue the SARS-CoV-2 signal from our wastewater, and these are the composite samples from one of the 2 major wastewater treatment plants here in Austin. And then plotted in red are the 7-day average of new cases within Travis County as a whole. And so it's very similar to the graph I showed earlier of the data from Connecticut, in that you can see the trend in decreasing SARS-CoV-2 RNA in wastewater much before you can see the trend in decreasing cases within Travis County of COVID-19. And as we're expecting, as winter starts coming in, people are spending more time indoors, and other factors, as we're expecting to see a second spike in COVID-19 cases, what we're hopeful to see is that we can predict and give kind of an early warning system of, hey, cases are going back up. We should probably look into going back to more strict social distancing policies or having more educational outreach programs to make sure people understand that it's really important that we limit the spread of the virus again. And even further than that, once we do get a virus, I think that, or sorry, a vaccine, then I think that this data will really be impactful in determining where are quote unquote hotspots of the virus. So I'm just going to go over some steps for moving forward. Like I said, we've been doing this deep dive and really being sure that we're getting a representative signal. But moving forward, we really want to expand and make sure we're tracking that temporal trends in cases, the canary in a coal mine, if you will. We also want to enhance our model. So there are a lot of factors that influence what SARS-CoV-2 RNA concentrations we're seeing. And those can be anything from the travel time, how much SARS coronavirus 2 is shed in wastewater. And we really want to build a really cohesive model to ensure that we're tracking our concentrations with the case numbers. We want to include routine monitoring of wastewater within the UT Austin campus. And this gives us a very narrow look and can tell us, even within a dorm building, what kind of signal are we seeing. And just giving you the early warning to say, hey, maybe you should invest in some more individualized testing in specific neighborhoods or buildings even. And then finally, I think the thing that is most relevant to the sensors group is we would love to develop a device that could take some of those really unwieldy steps, including sample collection and RNA extraction, that would do it automatically for us and would represent a proportional amount of RNA to the number of virions that are passed by within the treatment system or within the wastewater conveyance system. And thank you so much. I do have a few acknowledgements. The whole Canary team, Austin Water has been incredibly supportive of us. And thank you all for listening.
Okay, thank you very much for your amazing presentation. Before the questions are coming in, and I have a question about the stability of the virus in the wastewater from your presentation. It sounds like there's a significant time lag between data collection analysis. So probably the viral population will change a lot. So how can you obtain results that are closer to the real case?
Right. So I think that question kind of breaks into two parts. It does take us a while to do data analysis, and that's something that we're working with the Texas Advanced Computing Center to really speed up that analysis so that we have that data when our results are applicable to the current case number. And then in terms of making sure that we're getting the virus that is being released into the sewer shed at the time that it's being released, we're actually planning to do several studies that analyze the stability of the virus within the conveyance system. So that's a great question. But I think because we're tracking the changes in concentration, that kind of normalization takes care of itself if we're simply tracking trends.
Yeah, that makes sense. Thank you. Do anyone have any questions about the talk? Okay, there's a question coming in.
Yes. So the question was, so right now you sort of pick up the samples on a daily basis and do the experiments within the lab. Do you think the technology can be downscaled and installed within the wastewater? So we would love to do that. And I think when I was speaking about moving forward, that's something we would love to be able to do and install that technology within the wastewater system instead of having that really bulky step of transporting and then analyzing and processing the wastewater within a lab. But I think that's going to take a lot of development. But I think once that technology is created, I think it would be applicable to new viruses and diseases that we see moving forward too. So I think it would be a great investment to try and create that technology.
All right, I have another question about the waste water flow. During a raining day or wet season, more water comes into the sewer system, which will probably dilute this concentration of virus. So wonder how you deal with the uncertainty of the wastewater flow.
Right. So I think one of the benefits of looking into the solids specifically of our virus is that we're not necessarily as concerned about the liquid content, but we are looking into some mathematical normalizations in terms of understanding what we're what our virus concentration is in units of. And so we've looked into a variety of things. We're thinking about moving forward with normalization to E. coli. So E. coli is a bacteria that we shed through our waste, and normalizing to that content might normalize that the concentrations we're seeing. We're also looking into normalizing by the total suspended solids content, so the amount of particulate method that we particulate matter that we see within our sample.
Okay, thank you. There's another question from our audience. Right now, people probably use much more soap than earlier. The soap also goes into the same waste. Does it affect your ability to pick up and detect the virus?
Yes, it does. So soap is a surfactant, and surfactants can disturb the membrane or the package that the viruses come in. And so there is the potential that it could impact the viral signal that we're seeing. However, I think that typically with the soap concentrations we see compared to the amount of waste that's being put into the wastewater, it's not as much of a concern, especially when you're getting that composite flow. It might be more of a concern when you're getting the grab samples that I mentioned earlier, because if you happen to grab a sample while there's somebody doing their laundry, there's going to be a pretty high concentration of soap. But I think in a composite sample, that's less of a concern.
Okay. Due to the time constraint, we'll take one last question. How do you plan on implementing the warning system to indicate spikes in the COVID cases?
Yeah, that's a great question. I think that as we have really honed in on our methods, now that we're moving towards ramping up our testing procedures, I think it will be much easier to automate and really get that high throughput of processing samples. And then I think this is going to be a huge collaboration effort with people who are in policy, with people who are, influencing the testing. And I think really having those open lines of communication will be really important, both within individual the university, individual organizations, and within higher levels.
Okay. I would like to thank you again for your great presentation. Yeah, and I wish you the best in your future work. If your question is not answered or you have further questions, please email us and we'll deliver your questions to Emma and get back to you soon. And I'd like to thank our audience as well for joining us today. And our next talk is, sorry, give me a minute to share my screen. Okay, our next talk is mitigating exposure to indoor air pollutants from bleach and peroxide cleaning events given by Daniel Blomdale on October 9th, 2 weeks from today. Daniel will talk about their world-leading mass spectrometers. So please remember to join us next time. We're very much looking forward to seeing you again.
Thank you.